Our next presentation is the Lindau lecture that Countess Bettina Bernadotte mentioned yesterday in her welcome address. And it will be given this year by Aaron Ciechanover, who won the Nobel Prize in Chemistry in the year 2004, together with Avram Hershko and Erwin Rose, for the discovery of ubiquitin-mediated protein degradation. And today he will talk about the personalized medicine revolution. Are we going to cure all diseases, and at what price? Aaron, the floor is yours.
Well, good morning, everybody. I would like to start with two personal comments. One is that lecturing in Lindau is much easier for me. And the reason being that chemistry and biology and medicine are tangible. You can see it. You may not see an electron, but you can follow an electron in a hydrogen bubble cell. You may not see a protein, but you can sequence it, you can crystallize it, you can label it and put it on a film and see it. While your world is the world of abstraction, and for me abstraction is very difficult because when I close my eyes, I have to see things. I find myself completely detached. I cannot do anything. I'm lost, totally lost. So now I have the task of bridging between the two worlds, and I think that part of this difficulty is coming from my education. I am not a scientist. I am a physician. And I'm even the wrong physician. I'm a surgeon by training. And surgery is all about seeing and doing. There is nothing about imagination. Of course, you need to decide quickly and to use a lot of adrenaline and so on, but it's the complete extreme of what you are doing. So thinking about what to talk to you today, I decided not to talk about our discovery on protein degradation, which is fascinating and applied to medicine in a big way these days, but rather to talk on my other side, on medicine, and mostly about the information revolution in medicine, which is represented, I think, in the best way in the personalized medicine revolution. And we shall see how it goes. So whether we can build a bridge, basically it's a necessary bridge because there is no medicine today without informatics and without interpretation of the informatics. The second comment that I want to personal comment is looking yesterday at the three people that passed away from your area of this field. Of course, I was captured by the photo of Mariam Mizarkani and read the many obituaries that were written in her memory in recent months. I must admit I didn't understand much of what she did, but seeing the torch of life of a young person extinguished in front of your eyes and being completely helpless gives us a sense of where medicine in the extreme cases these days. The complexity of diseases that deepens and deepens as we look at it, and the fact that we are very much helpless in many diseases, in many malignancies, brain diseases, we don't have even a clue to the mechanism, not to say the therapeutic is frustrating on one hand, but on the other hand, I hope that via the lecture you'll see that there is a ray of hope and we are starting to take the right way. But this is, seeing a young person dying in front of your eyes is the biggest frustration that the physician, not to say a human being, can face. So let's talk about the revolution of personalized medicine. This is my city, the city of Haifa. It's a beautiful city rolling on the east side of the Mediterranean. You can see the Baha'i garden. The Baha'i that were persecuted in Iran came to Israel bearing their leader under a beautiful temple and converted the whole site into a beautiful rolling gardens. It became a UNESCO heritage site. And the medical school where I am is just on the sea here, in this very point. I'm looking from my life directly to the sea to the north, and it's just gorgeous. So personalized medicine revolution, what is it? Why revolution? Why now? What it entails? How it began? Where it goes? So of course, one point to look at it or to start it is the lifespan. If you think about people living 100 years back at the turn of the 20th century, the average lifespan was about 50, 55. And people didn't die of cancer because they didn't live long enough to have cancer. They didn't die of heart attacks because they didn't live long enough in order to get heart attack. They didn't die of Alzheimer. The three main killers of modern medicine: heart, vascular system, malignancies, brain. They died of infectious diseases. The woman died during delivery. Out of 10 children in the family, 5 could survive, and the 5 were used mostly as a working force in the family. Forget about the families of 1 or 2 children these days that we are teaching pianos and computer sciences and we care very much about their education, just merely 100 years ago. When we go back 4,000 years ago to the Greeks and to the Egyptian time, and Chinese, that were isolated from the world, the Indians, people were living on the average 30 years. So this was the average lifetime span. So it took 4,000 years to expand lifespan from about 30 to 50, 55. And then it took 1 single century, about 2.5% of the time, to jump another 30, 35 years. Now the average lifespan in the Western, in the OECD countries, is about 82, 84, depends man, woman, changing. Japan is leading, 86 even for women. So 1 single century, we were jumping more than in 4 millennia. And what is it? Of course, science and technology, in big way. It's understanding diet, it's vaccinations, it's drugs, it's X-ray imaging, sewage systems, sterility, whatever you like. Take all of them together, all were developed in this magic 20th century, and you have additional 30 years to our lifespan. And the curve is still going up linearly. The United States started to go down a little bit, but that's mostly due to violence, poverty in recent years, but mostly it's going up in a range of several weeks a year now, so 2 to 3 weeks a year we are extending our lifespan. There was a recent prediction in Nature that the upper maximum will be 115. People doubt it. Some argue that we may be able to go farther than that. And then, what about diseases? What about quality of life? The idea is not just to live, the idea is to live with high quality of life. Are we going to defeat diseases? Are new diseases going to surface that we didn't know before, like cancer and heart diseases and brain diseases were not known? Well, they were known, but nobody thought that it's an epidemiological problem. At the turn of the previous century, are new diseases going to surface in the future? So, are we going to cure all these diseases and mostly are we going to pay a price for it? And by price, I mean both dollars, which is going to be a problem, but also other prices that we may not think about, but I'm going to highlight them in a big way. So if we think of medicine, many diseases are preventable. As scientists, we don't tend to think about it. If I ask, because I'm teaching medicine to medical students, in my faculty, when I ask people what do they want to do when they will grow up, most of them want to be brain surgeons, heart surgeons, you know, all the hardcore prestigious. None of them want to be an epidemiologist. But if we think about it, epidemiology and prevention of medicine should be the way to go, because this is the most logical way and the cheapest way. Think about obesity, which is an epidemic in the Western world. This is a preventable disease. It's a psychological disorder. If you want to go to the extreme and think about people that consume 5,000, 10,000 calories a day, smoking, contamination of the environment, infectious diseases, you know, eradication of mosquitoes, the Gates Foundation that fight malaria and so on. So many of the big killers are really preventable by simple measures and by small investment. It's just our selfishness that prevent us from going into the right places, because people are people. They go after their behavioral pattern rather than after where they should go. But that's another issue of discussion of a big view of medicine. But let's go into medicine today. Medicine today is moving in 3 main directions. It's moving, of course, in devices. Devices are extremely important. Engineering, actually, it's not the physicians. These are the engineers, if we think about it, think about X-ray, the first Nobel Prize in 1901 in physics that went to Konrad Wilhelm Röntgen, looking into the body without invading it, magic. And then it held for almost 80 years, one technology, until it was gradually replaced by CT scan, MRI, and now CAT scan that can also look at soft tissues. The issue with X-ray that it was either bones or nothing. Now we can look at soft tissues. So devices, artificial valves, artificial organs, artificial joints, small cameras, drillers that can go into the arteries and clean up and drill out occlusions in the coronary arteries that feed blood into the heart itself. So technology on one hand. Then regenerative medicine, stem cells. The promise is still not there. Hopefully it will come. But replacement of tissues that are not functioning anymore. Think about diabetes, which is probably #1 disease, #1 epidemic in the Western world. These are non-functioning cells in the pancreas, in the Langerhans islet, that secrete insulin. And actually, if we shall be able to reprogram cells to secrete insulin, we don't have even to implant them in the pancreas. It doesn't matter. We can put them under the skin in the hand. They will secrete the hormone, and the hormone will go into the circulation and reduce glucose level. Parkinson is a little bit different because it has to be in the brain, because it has to secrete a hormone that is a dopaminergic compound in the place in order to balance for the activity of the cholinergic system. Because remember, in Parkinson, we lose control of our stability. We cannot write, we cannot cut our food, we cannot button up because there is a dominance of one system over the other. Think about even stability, the fact that you are sitting to now stable, listening or being bored to listen to me is a miracle. How is it? How does it happen that the muscles and everything is just in place and undertone? You are not relaxed. It's undertone. It's kept undertone. Otherwise, you will fall aside. So that's so tissue engineering and stem cell has a topological problem for some of the implantations. Like if we want to put a muscle, the muscle needs to be not only put in the right place, but also connected to the brain in the right way, so it will perform the right movement. But then there are tissues that have no topology, like blood, like insulin-secreting cells, and so on. So regenerative medicine in big is the second direction. And the third direction is, of course, the traditional one, drugs. And we shall focus mostly on drugs, because time limitations, we need to focus. So what happened to drugs? How drugs were developed? So drugs were developed, the first revolution, we are talking just the 20th century, because before the 20th century, everything was completely meaningless. There were drugs, there were physicians, the barefoot physician in China, all over, but the communication enabled us to made a major breakthrough, the ability to run clinical, objective clinical trials in different clinical centers in the world, not knowing what one center is doing, the ability to collect data together. It's all, actually it's not even the 20th century, it's the second half of the 20th century. So we are limiting our discussion mostly to the last 70 years or so. So the first discoveries were completely incidental. People discovered something, they didn't mean to discover it, but it turned out to be blockbusters. I don't have time to show you all examples. I'll show you just one example, and that's aspirin. Aspirin is a drug that is being taken in tons. It's probably the most used drug ever formed in the pharmacopoeia. Many of the faces here, of course, of the young researchers are young, but of the little bit more advanced, I'm sure that many of you, anybody is volunteer to tell me how many of you are taking aspirin daily? Oh, see, there are some hands, me too. It's really an unbelievable drug. Most people take it in order to prevent blood coagulation and heart attack. Some people like me take it in order to prevent cancer. It turned out to be one of the best chemo preventive drug. And it started about 4,000 years ago when the old Egyptians noted that when they are eating, when they are chewing the willow leaves, the willow bark's leaves that grew on the banks of the Nile River, the taste was very bitter, but it had pain alleviating properties. It remained like that for 4,000 years until at the end of the previous century, French and German scientists took the bitterness out by acetylating it. It's a very simple compound. It's acetyls salicylic acid, very simple compound. Anybody can synthesize it in the basement. And they took the bitterness away. And then two scientists, Eichengrun and Felix Hoffmann in Bayer AG in Germany, the father of one of them got rheumatoid arthritis, which is an inflammatory disease of the joints, which it's a paralyzing, debilitating disease. Again, people cannot do anything. They are painful. The joints are inflamed. And they wanted to relieve the pain of the father, and they read the literature, and they decided to make aspirin. And aspirin not only took the pain away, but took the inflammation away. So they realized that they discovered not only an analgesic drug, a pain-relieving drug, but also an anti-inflammatory drug. They went to Bayer, they convinced them to make the drug officially, and the rest is history. Now aspirin is being made all over the world, not only by Bayer. The patent expired years ago. Gradually what happened is, the two things happened during the last, I would say, 3 decades. So they started in the 80s and still going on. First of all, it was discovered that aspirin prevents blood coagulation. So it's a very good drug in order to prevent heart attack. What is heart attack? Heart attack is coagulation of blood over a cholesterol wound, over a plaque, over an atheromatotic plaque in the coronary arteries that lead blood to the heart itself. So the muscle of the heart doesn't get blood, and we succumb, we fail, we fall dead, or luckily not dead if the occlusion is not complete. Here you can see the patient suffering from the chest pain. It looks like President Clinton, but it's not. So this was the first attribute, the first characteristic. The second one was the linkage that was actually discovered 120 years ago by Paul Ehrlich here in Germany about the linkage between chronic inflammation and cancer. Now, people are misled to think that inflammation is something like, wow, I have an inflammatory throat, or I got wounded and there is pus. No, inflammation is a disease that many of us are suffering without knowing, liver inflammation. You know, many of us, millions and millions of people in the world are suffering from hepatitis, from the virus. They don't know even, and gradually the liver is phasing out and losing its function. Chronic intestinal inflammation, breast, prostate, and Paul Ehrlich predicted that chronic inflammation, when the tissue is all the time stimulated, will lead to cancer. The mechanistic linkage was discovered in the recent decade, or the last 15 years, and it turned out that if you prevent inflammation, you can prevent cancer by just cutting this vicious chain in the middle. And the American Cancer Society declared aspirin is the best chemopreventive. Why all people don't take it? Because it's a drug, and drug cause side effects, bleeding, gastrointestinal bleeding. So it's not recommended en masse. This is always the double-edged sword of recommendation of drugs to people all over. But it is recommended only to people with family story or with high susceptibility from whatever reason to take it. But many people do take it because it's available on the shelf. You can buy a year's supply in $5 in CVS, Walgreens. I don't know in Germany what are the pharmacy chains. I'm more familiar with the American ones. Anyway, look at this, serendipity, complete serendipity, no investigation, no billions of dollars invested. The same story is antibiotic. I don't have the time to go into antibiotic, but the same story is for antibiotic. That's Fleming. I will just show you the serendipity in the discovery. Fleming was a microbiologist, and microbiologists are using Petri dishes that have agar, that have a nutritious medium in order to grow the bacteria. So this is the bacteria. This white smear are the bacteria. So he left by mistake a Petri dish open on the desk and went to a vacation. It was the summer, August of 1921. And when he came back, he saw this white spot. Now, the white spot is a spore of a fungi that is in the air, even now here in the air, that fell on the agar plate and grew because there is a very nutritious media. So the spore kind of proliferated and made the colony of the fungus. But Fleming noted that there is a halo between the bacteria that should have grown all the way to the smear, to the border of the smear, there is a halo here between the colony of the fungus and the bacteria. The bacteria cannot reach, cannot touch the colony of the fungus. And he surmised that the fungus secretes a material that he called antibiotics. Anti against bios, life. Biology is the profession, the area of life. He was not a chemist. It took another several years to Ernst Chains and Howard Florey to purify penicillin, and the world has changed forever. Since then, streptomycin came by Waxman. People realized that other fungi secrete other antibiotics, and then synthetic antibiotics, and so on and so forth. So complete serendipity. The guy was just looking at the dish. He didn't he didn't investigate, he didn't know anything. So two major drugs came by serendipity. But we cannot rely on serendipity, of course, not nowadays, to develop drugs. And we had to move to a better system. And the better system, the second revolution, which we are still into, is the high throughput brute force screening. Chemistry evolved side by side with biology. And what happened in the two sides is that in biology, biologists managed to develop disease models. Either in growing cells, I can isolate the cancer cells from patients and grow them in the dish, or in animal models. I can manipulate now animals in order that they will develop disease. Let it be Alzheimer, let it be, of course, cells cannot imitate Alzheimer, or cannot imitate depression, or cannot imitate autism. So some diseases can be imitated in culture cells, and some diseases can be imitated by animal model as faithful as they are. This is a caveat, as faithful as they are. So this is on the biology side. On the chemistry side, chemists were able to synthesize millions of compounds, and on the synthetic pathway, they left behind different intermediate materials, because you move from one material to another until you get the final product. So millions of compounds were accumulated. Many of them, all the intermediate materials are useless, even the end products not always being used. And people say, let's gather them in families, let's put them in families, and if we have a model, let's try to give the animal or the cells this million compound one by one. We don't know the mechanism, we don't have any hypothesis, therefore I call it brute force, and one of them will do it. Why? Because evolution is limited. Because the proteins have so many shapes, and the mutated proteins gain so many shapes, and one of the compounds will fit in to modulate the bad activity, to inhibit it, to accelerate it, to do whatever. The assumption turned out to be correct. If you are trying millions, and then, of course, it led to the development of lots of technologies, robots that are going to the incubators, take out the compounds, cells that are growing, bringing them together. And of course, computing came into the story, because once you put the compound and you let the cell grow in presence of the compound, then you take them to the microscope. So artificial intelligence software was developed in order to look at the cells and to say which one has effect, which one, to learn the shape of the cells, the division rate, and so on and so forth. Many parameters were put into this relatively simple, I assume in your eyes, software, and then you have a lead that does something, and then the lead is being taken further to further experiments, toxicity, human trials, and so on and so forth. And many very good drugs came out of it. And the good drugs, I'll bring you one of them, statins. Statins are cholesterol-reducing drugs. It's essential not to have high cholesterol level. It leads, again, to this yellow plaques. This is for sure not President Nixon, so President Clinton. So you see here the yellow fatty plaque accumulating in the coronary arteries and threatens to occlude the whole space. It's like rust in your pipelines, of water pipelines in your home. And then on top of it, the plaque also creates a wound on the surface. The wound attracts blood clotting, and that's the heart attack story. You will always say, oh, the guy was jogging and he was healthy, and all of a sudden he dropped dead in the streets while jogging. Well, he had the disease for the previous 20 years, but it was not diagnosed. He had this crawling occlusion coming up and up and up. So statins, it's Akira Endo, a Japanese a scientist working in a pharma company. He screened a library of natural products, fungi, fungal products, pine tree products growing in Japan, and one of them did inhibit the biosynthesis of cholesterol, and statins became a big blockbuster. Last year's sales were more than $40 billion by different American companies. I'm sure that if I'll ask you to raise your hands, Anybody uses statins? Look, just have a look. And it's all the front rows. Of course, the rear rows are still not there. So statins are magic drugs, not only because they are reducing cholesterol, there are now claims that they are also preventing prostate cancer, that they are also preventing Alzheimer. I will not go into all these links and the clinical trials that are still going on. So statins by screening. Now screening, unlike serendipity, is a good way to go. Let's screen and screen and screen and screen until we'll defeat all diseases. What is the disadvantage? The disadvantage are the models. We are screening mice. Mice are nice, but they are inbred animals. We turn the light at 8 o'clock in the morning in the cages. We turn it off at 8 o'clock in the evening. We feed them on chow purina. These are not human beings. Human beings are vastly different from one another. Not only that we are men and women, we are much more dissimilar to one another than we are similar. Think about blood infusion. We cannot even transfuse blood to one another. There are 8 different types of blood, right? A, B, O, plus, and minus for each. Not to say a kidney transplantation or lung transplantation, this is 1 to 10 million. We need to be HLA fitting on the immune system. So we are very different from one another. We are different in diet, we are different in genetic repertoire, we are different in the sun, we are different in every single aspect. So the model is the wrong. This is one issue, that we are different. And then, to our naivete, until about 30 years ago, we thought that cancer is cancer, breast cancer is breast cancer, it's a lump in the breast. Now, when the ability to sequence the DNA came at the late 90s, and we were able to look at it, it turns out that breast cancer is not breast cancer. These are 20 different breast cancer. One is derived from a mutation in the progesterone receptor, and one in the estrogen receptor, one in the EGF receptor. One is now called triple negative. It's kind of a basket to which we throw all the other that are not the other 3 that I mentioned to you, but in this basket they are hidden probably about 15 or 17 more. And what we call the disease was basically wrongly nomenclatured as a phenomenon, as a description. But molecularly, it's very different. Think about Alzheimer patients. One will deteriorate like this, and one will deteriorate like this. Think about heart attack. Some people will die on the spot. Some people will survive 5 heart attacks. Think about any disease that you want. They're all variable. They're all variable. And we knew about it, but somehow we closed our eyes. We said, you know, I remember myself as a young medical student, this was not an issue even to be discussed in the medical school, because we didn't know much. And we said, this is like, okay, we don't know what it is. But now we do know what it is. And we now have the tools also to stratify the diseases. And this is the essence of the revolution of personalized medicine. So now let's just go briefly into it before we shall discuss the problem. So it's not only personalized, President Obama called it also precise. Yes, personalized, precise, it's the same. It's not personal in the sense that there will be a drug for any Mrs. Elizabeth Changju or whatever, but there will be drugs for groups of patients that have the same molecular mechanism that is driving the disease. This is the idea. And in order to diagnose it before treating, we shall have to first unravel the mechanistic reason behind the disease, the pathogenetic mechanism. So this is the idea. Because for now, it's the opposite. You come to the clinic, and there is a breast cancer, and then we don't care about the name of the patient, we don't care who she is or who he is, or doesn't matter. If the breast cancer is isolated, we take it by surgery. If the breast cancer is already spread by metastasis, we start with chemotherapy, and then we go to surgery if possible. So we don't care about the carrier of the disease. The mirror of medicine until now was shining towards the diagnosis of the disease. Now, it will flip by 180 degrees into the disease in the context of the patient. So this is the major essence of this revolution, and it's critical, it's fundamental. It's going to change the face of medicine in a very big way. So it's personalized. By knowing what the patient has, we are not going to limit ourselves to the disease. We are going to sequence the entire DNA, because you are not going to sequence the DNA of the cancer of the patient. You are going to sequence the entire DNA. And then it's going to be predictive, future. which tells you already the first issue here, the first bioethical issue. Do we want to know what we are going to have? Don't we want to know what we are going to have? Does it belong to us? Does it belong to the insurance agency? Does it belong to the government? Do our spouses want to know? I'm just hinting to you about it. So it's going to be predictive. It's going to be preventive in many ways, because if we know what we are going to have, even now, but for sure in the future, we shall be able to prevent it. So you see, it's highly implied. It's not just, okay, let's do the DNA. And most importantly, it's going to be participatory. I remember the days that I was a young medical student in the 60s, that medicine was a very patriarchal type of a profession. Trust me, I'm your doctor. Don't ask too many questions. You are a lay. Gone are those days. And they are gone. Internet, of course, first and foremost, legal suit against companies, against physicians, and the complexity of the problems. The physician is changing his role or her role now to become a professional consultant, kind of an advisor, but not a decision-making, unlike it used to be 50 years ago or even 30 years ago. Completely different role. And we are implementing it already in the medical education. We are putting the physician on the sidetrack, and we are putting the patient and the family and the colleagues and what the patient have to think about him or herself in the on the center stage. It's a very complex revolution, and somehow difficult also for doctors. Doctors are unique type of creatures. There is always a joke about the difference between God and doctors, that God doesn't think that he is a doctor. But it's a joke, but if you are teaching in a medical school, it's not such a joke. But anyway, so it's going to be participatory, it's going to be a dialogue, and we are going to touch on all that. I want to give you just one example about this type of medicine, so you will have a clue. That until now, really, medicine was, in a way, very primitive. Actually, it's still very primitive. It was like what I call the pajama. Pajama is something, unless we are very fancy royals, is something when I walk, well, in Israel, you don't need pajamas because of the weather. But if you're living in Minnesota or in Boston or on the North Pole, you do need something to protect yourself from the chill of the night. But pajama, you know, you walk to a store and you ask, what do they have? And they tell you that they have small, medium, and large, right? You don't really measure it. You don't go to a tailor to make your pajama. While a suit, you do go to a tailor. So until now, medicine was kind of a pajama, one size or, three sizes fit all. And what we are going now with medicine to the tailor. We are moving from the pajama to the suit. And the reason is exactly the same, that when we are having a group of patients that are diagnosed at the same day with a certain disease, and if we give them the gold standard current treatment, and we tell them, now you're starting the road, and when one of the patients are asking us, or the doctor, tell me, am I going to make it through, or amn't I, or I'm not going to make it through, the doctor tells the patient 70%. Well, for some diseases we know that they are deadly, like brain cancer or most of the pancreatic cancer, but 70%, 80%, 10%, doesn't matter, it depends on the disease. And then the patient keeps on asking, and he or she tells the doctor, but I have a name, and I have a family, and I have an address, and I have an ID card. What is going to be my fate? The doctor tells her, I don't know. And what he adds, if he wants to further elude on the problem, he said, listen, I am 70, 80 years old. I've been sitting in this clinic for the last 5 decades. I've seen 2,000 like you, and the statistics is like that, and therefore I'm giving you a number. But the number is not sufficient, not for the individual patient. But that's what medicine could have done these days. We didn't know whether the cancer will spread, or it's spread already. We didn't have diagnostic tools. We didn't know anything. We knew statistics. That's what we knew, based on, of course, clinical data. If this cancer is not spread or not metastasized or whatever, or if the deterioration of the Alzheimer patient by cognitive test is going along this line, the assumption that it will continue to go along this line and you can make a trajectory. But otherwise, the tools were awfully primitive. So patients were split, and we could know only after 5 years or 10 years of what's happening when they split into those that were unfortunate, to those that were fortunate. But we didn't have any predictive tools. I think that now, with a precise medicine, we are going to have much better tools, much better statistics, and so on and so forth. So let me give you just one example, so you will have, it will come back to the beginning of my talk. You will see it. I mean, seeing is, I don't know why seeing is believing has become such a strong statement for me, for sure. Seeing is the only way of believing. So here you have two patients, patient A and patient B with breast cancer. So you see the cancer, you see the island of the malignant cells in the biopsy. You see here the islands. And you can see that this island is not stained with an antibody. So the brown is staining with an antibody. It is against the mutated estrogen receptor. While here we try to stain, but no stain. It remained native. So this woman, woman B, has a mutated estrogen receptor, while woman A doesn't have it. She has something else because she has cancer. Therefore, woman A, woman B can be treated with tamoxifen, which is a drug to counteract the activity of the mutated estrogen receptor, while woman A, treating her with tamoxifen will be a waste of money, but mostly a waste of hope. Why to treat her with tamoxifen? It's like giving her water. So she should treat it with something else. She may be treated with Herceptin, because she may have a mutation in the EGF receptor. But now it's all available. We can we can take the sample, and we can sequence the DNA and find out what she has. And then, if she has a mutation in the EGF receptor, then we can treat her with Herceptin. By the way, Herceptin made a history because it's the first biologic drug. It's the first, not small molecule, but an antibody against a surface molecule on the surface of the cancer. Or she may have a mutation in the progesterone receptor, or unfortunately, she doesn't have either of the three, and she will be classified as triple negative, which is the most aggressive one because we just don't know what to do nowadays. So this just gives you a little bit of a glimpse on the stratification that there is no cancer of the breast anymore. There is estrogen and progesterone and EGF and NF-kappa-B and FOS and JUN and MYC and all the others. And from now on, the diseases will be stratified according to their molecular cause rather than according to the visual lesion that can be seen either in X-ray or by palpation or by whatever. This is very primitive, we should all admit. So, it's true not only for medicine, for drugs, but it's true also for side effects. Still a few minutes. But for side effects are a major problem. About 4 of 6 of the patients in American hospitals are dying because of side effects of the drugs. And this is not because of negligence of the physicians. Most of the side effects are not known a priori. Well, I'm sure that you know that if you go to a hospital, the physician will ask you, are you sensitive to penicillin? That's the only thing that we know. Or is there any sensitivity in your family? It's meaningless. We don't know. Every drug has side effects. Every drug. Some of them are totally unexpected. And worse, we don't know it a priori. We just don't know it a priori. And they are all derived from the same very reason, mutations in the DNA. So once we shall start to collect patients with the same side effect, with the same reaction to the drug, we shall be able to go back into the DNA and to say, yes, the mutation in this gene is the cause, and to predict a priori. So you see, it's not going only to drugs, it's going only for what we call ADR, adverse drug reaction, which is a major, major issue in medicine. So it has many, many implications, this revolution. But let's go to the what we call the price. And the price is, by the way, I don't want you to get out of this talk with a naive thinking that DNA is the whole story. It's far away from being the whole story. DNA is the very base. On top of the DNA, there is RNA that is being transcribed out of the DNA, and mostly on top of the RNA, there are the proteins. We are running the machines. I always divide life into 2 dimensions. The horizontal dimension is reproduction. Of course, it's DNA. But the vertical dimension, and the horizontal dimension is the proteins. We are running on our proteins. The muscle, the hemoglobin in our blood, the vision, the receptor, everything, the pumps on the cells, it's all proteins. So we have DNA and we have RNA and we have proteins, and then we have small molecules. And side by side with genomics, with our ability to read our DNA, evolved different fields that are all under the umbrella of omics, which is wonderful, which is transcriptomic for RNA, and proteomics for proteins, and metabolomics for small molecules. And at the end, we are going to have a whole profile of the patient in front of us and a very, very accurate picture of what's going on. And it's going to be dynamic. We shall be able to monitor it as the patient is moving along the treatment, whether the treatment helps or doesn't help, and so on and so forth. But let me go to the price of this revolution. And the price is going to fall into two sides. It's going to be technical, of course, because not all diseases are derived from one mutation in one gene. Many diseases are what we call polygenic, mostly psychological diseases, but also some metabolic diseases. But let's forget about technology because of time limits. I want to go mostly to the bioethical issues. And the bioethical issues are mostly pertaining to what you are doing. And that's data collection and privacy. I'll give you an example. I'm going to the emergency room because of chest pain. Chest pain is always an indication of heart problem. And the doctor rules out heart attack. And he comes cheerfully to me and tells me, Aaron, you don't have a heart attack. But then he comes and he said, I have something else to add to you because now sequencing of the DNA will become a routine. It almost becomes a routine now because from the first genome that was sequenced for $1 billion in 7 years, now it takes about 2 hours and about $1,000. So the technology was changed. We are going to solid-state sequencing. Computers are coming to our aid. The whole things have changed dramatically, and it will become routine. And he said, you have a mutation in ApoA4 gene that carries with it high susceptibility to Alzheimer's disease. Well, high susceptibility is likelihood. It's statistics still. We don't know yet and what we can do about to prevent it. So I'm under a bombardment. I am under a bombshell in the emergency room. Alzheimer. I came for a chest pain. I don't have heart attack, and I'm walking out with high susceptibility. Now, what I'm going to do with it? What this information worthwhile? To whom do I talk? Do I talk to my wife? I assume that yes. Okay. Do I talk to my children? They may carry the gene. What they are going to do? They are still young. They have not been married yet. They are in the college. They are just married, fiancé, children, beginning their families. Do I talk to my employer? Will he fire me? Do I talk to my insurance agent? You know, insurance policies are being renewed yearly. Why should he renew my policy if here he has a patient with 80% likelihood of Alzheimer's? So I paid for, all my life when I was healthy, I paid for 40 years health insurance, and now when I need it, out I go. So think just about what is the significance of this information, to whom it belongs. Should the doctor hide it for me? Should he tell me only the information that there is something to do about it and keep the rest for himself or in the files of the hospital? Are the files of the hospital safe enough or they are not safe enough? Will the government would like to know what the citizens have? Would the army, the military would like to know what people have in order to draft the most intelligent and most fit soldiers to the military? I mean, it's going everywhere. I want to end up, because time is really running out, with one photo that will give you a glimpse into it and will show you how things are going to change. You see, this is issue of genetic privacy and what is the meaning of privacy. Privacy until now was something that I think belonged to me, but it's not necessarily belong to me because there will be other agencies that may be interested and there will be legislation and so on, and I can bring you also personal information that you are forced to give your information just because of humanitarian reasons. It has nothing to do with outside forces. It has to do with helping with the kidney to my brother that is dying of chronic kidney disease, and whether I do carry the gene myself or I don't carry the gene and I can help my brother or my sister by donating the kidney. And so it's penetrating every layer of life. It will be very difficult to put delineation marks or lines of delineation there. So let me, you see, everything is going, wow, what's going on here? We are at the beginning. But okay, let me go to the very last one. But you see, that I'm afraid to play with it, because then, you see, it's all genetic privacy, do prepare for big genome leak. Everything has to do with keeping the data. But I want to end up with the photo of Angelina Jolie. And Angelina Jolie came a few years ago with a story. Actually, I remember exactly the dates. It was the middle of 2012 and then the beginning of 2013, that she's going to undergo a first operation to remove her breast, and a year later to remove her ovaries. Can you imagine? The symbol of womanhood is going to remove the ultra symbols of womanhood. I mean, think about it in a movie industry type of a thinking. And the reason is that her mother died of, or was going to die at that time of breast cancer, and her aunt, the sister of the mother, died before of ovarian cancer. And she suspected that she's carrying a gene that is called BRCA1. I am attached to BRCA1 because it's part of the ubiquitin system that we discovered aside, you know, so this brings me the linkage, but I'm not going to talk about it. And she suspected that she's also carrier of the gene, and therefore she's carrying in her body a ticking bomb, and she's going to develop with high likelihood the disease. 80%, here is the end, that she's going to by 80% get breast cancer and by 40% get ovarian cancer. And breast and ovarian cancer are completely different ball games clinically. Breast cancer is manageable, well, not for all women, unfortunately. Ovarian cancer is deadly. It develops in the abdomen. It takes a long time until the woman complains, and once, typically, in 99% of the cases, one is discovered, it's over. So she has undergone the test, and she was found to be positive, and she decided to go step by step and to do the operation. So she did a great service to women worldwide to raise alertness, and it depends on the culture in the country. In Israel, it didn't move anything. Women are women. They're conservative. They don't care. Even if they carry 2 mines on their back, they will keep on running. In America, it was different. It's very interesting to follow the response in different countries, the culture, tradition, religion, whatever. But what about her daughters? What about her daughters? Well, she adopted, but what about the daughters of other women? Should she tell them? Again, the same story. Maybe they are carriers of the gene. What they are going to do, to tell the boyfriend, to tell the newlywed husband, to tell the fiancé, to tell their children. What about it? Now, it looks frightening, but let me just conclude the talk in really big ray of hope. Because if you think about the history of science, science have always been posing a question, putting the problem, and then solving it. We cannot solve a problem without putting the question ahead of time. And this is the same situation here. It's not different. We need to understand the problem in order to solve it, and there is a solution already behind the corner. It's not a magic solution because it has many problems of its own, but that's a technology that is called CRISPR-Cas. It's a gene editing technology. I'm sure all of you heard about it. It's already being in use for limited purposes, by which we can take a mutated gene, cut it away, and repair it. So in the case of breast cancer or ovarian cancer, these are 2 removable organs. So Angelina and her allies were lucky because they can be subjected to complete total surgery. You can remove the breast, you can remove the ovaries, and you are removing you are taking a preemptive bomb, you are taking a preemptive step in order to remove a bomb. But in other cases, we shall have to repair the genes. And this raises other ethical problems. You see, we are never solving the problem. That's the beauty of science. We shall never get bored, because new problems will come. What is a disease? Some women will say that they want to have tall children that will be basketball players, and they will discover that in their uterus they are carrying little children that are going to be disastrously computer scientists. So, and they never dreamt of such a nightmare dream. So you see, I'm exaggerating, but the solution will come, but new problems will come along with it. The definition of disease will change, and we'll have to cope with it. And the last very sentence is that coping with this complex problem should not be put on the shoulders of scientists like you and myself. We should be, again, the advisors. We should be the professional side. This is a societal issue of clergymen, legislators, historians, psychologists that will all sit and look into it. We are already into the problem, but unfortunately, society haven't started even looking into it. Thank you very much.